everyone. Today we are going to discuss the very basics of genetics. This will help you to understand what genetics, what heredity is all about, what genes are, what alleles are, and what were the Mendel's experiments that led to the development of various laws related to genetics, and clearing the concepts, how our blood groups are associated, how we can tackle the issue of blood groups, how we can understand how blood groups are formed. So, all these topics would be covered in today's discussion session, and to begin with, we are discussing about the heredity, which is like the biological process through which we all know that genetic traits and characteristics are passed from the parents to their offsprings. And this process is fundamental to the study of genetics and plays a crucial role in development and evolution of living organisms. And we know that there is some kind of variation from parents to offspring, and the degree of variation would let us know that how offspring would be different from the parent, and that all the codings are involved in form of genes within us. And it refers to the transmission of genetic information from one generation to the next, explaining how the traits such as physical appearance, behavior, susceptibility to certain diseases are inherited. For example, we have all heard that if our parents suffer from diabetes, and there are chances that the offspring or the next generation will also have the chances of having diabetes. And not only diabetes, there are multiple other diseases which are associated with hereditary disorders, and for example, some cancers are also associated with our gene makeup. So, all these things are inherited from the previous generation to the next generation. And then genes. First, we will understand what are the basic concepts of heredity, including the genes, alleles and the chromosomes. The genes are the basic unit of heredity, which are segments of DNA that code for a specific trait, and they are inherited from both parents. If we talk about humans, because we are diploid organisms and we get the gametes from both the parents and thus the genes from both the parents. Chromosomes. Chromosomes are the structure within the cell that contain genes, and human typically have 23 pairs of chromosomes, that is 46, and one set is inherited from each parent. That is, we receive 23 chromosomes from our mother and 23 from our father. So, we are mixture of both of those traits. And sometimes you must have heard that you look like your father, you look like your mother, because how their genes are more dominant from which parent, that will affect your physical appearance as well. Then we have alleles. Alleles are the different versions of a gene that can exist. For example, a gene for flower color may be a red allele and a white allele. That means the gene is same, but it has different variations or version of that particular gene, which causes a different physical or genotypical trait. It may be a color of flower, color of your eyes, or size of the flower, or the size of the stem in terms of plants. So, there are different alleles that are present on the gene that can ultimately tell us what kind of genotypical makeup would be and what phenotype would it be dominating. So, this is an example, or this is the pictorial information where you can identify different alleles on the same gene. So, if the same allele is present on both the chromosome, then it would be a homozygous alleles, but if different alleles are present, then it would be a heterozygous allele, that you can see on the figure at the lower end, where AA and the different alleles have been shown by a different symbol, capital A and small a. Combination of capital A and small a would be a heterozygous allele, and combination of same alphabet would be a homozygous homozygous allele, like capital A and capital A, or small a and small a. The coloring has been shown to differentiate between the different kind of alleles that can be present. So, the genetic composition or the genetic makeup of a cell is known as a genotype, or an organism is known as a genotype, and the physical appearance of the organism is known as the phenotype. And by looking at the phenotype, you can't identify its genotypical makeup. and genes have two pairs of alleles. If both of them are of the same then they are homozygous, as I have shown in the previous slide, and if they are different, then it would be a heterozygous allele. So, a true breeding line is one that have undergone continuous self pollination, shows the stable trait inheritance and expression for the several generations. If it is all been constant, no changes, and the pollination has been done within itself the plant, then it would be a true breeding line, in that the alleles would be homozygous and everything would be passed on as such in a stable format to the next generation and continued to the several generations ahead. And by simply looking at the phenotype of a dominant trait, it does not possible to know the genotypic composition. We are studying these key concepts at the initial stages so that it will help us to understand the rest of the concepts in the hereditary or the Mendel's experiment, which would give us the detail of identification process of phenotypes and the genotypes and what expression they are creating within the organism. So, Punnett square. It is a graphical representation to calculate the probability of all possible genotypes of an offspring in a genetic cross across different generation during the formation of zygotes. So, it will tell us whether the zygote would be of which kind, and it is a probability square which tells us which zygotes can combine and which kind of phenotype can be generated. It is called Punnett square because it was developed by a British genetist, that is Ragland Punnett. Now, we will talk about the Mendel's experiment. Mendel was known as the father of genetics, who gave all the concepts to the genetics. And before that, we should understand that it is not that before Mendel, because Mendel conducted experiments during late mid 1800 BC century. For the 7 years he continuously experimented and worked upon the laws for genetics. But it's not like the situation that before Mendel's experiment we didn't knew anything about the genetics, although I agree that there were some terms that were came into existence or came into knowledge post Mendel's experiment, but humans knew from as early as 8000 to 1000 BC that one of the causes of variation among the offsprings was actually hidden in sexual reproduction, and humans exploited the variation that were naturally present in the wild population of plants and animals to selectively breed or select organisms of their choice that possess the desirable characteristics they wanted. So, it has been long known that there is kind of possibility of variations, and those variations can be selected to create an organism or to develop an organisms with selective or with desirable characteristics. And the very common example of artificial selection and domestication is the domestication from wild cows, and now what is known as Sahiwal cow in Punjab, that has been worked upon very long back through selective characterizations of organisms through humans before Mendel's experiments. So, Mendel's experiments were basically done with pea plants over the period of 7 years, focusing on 7 particular traits, and they revealed the fundamental principles of inheritance and in a documented way. Further, Mendel was the one scientist who brought mathematical expressions or mathematical calculations in biology. So, the probability testing and the relationship of biology with mathematics in genetics was conducted by Mendel. So, Mendel chose pea plants by some sativum for his experiments. There were particular reasons of selecting this pea plant for his experiments was that because they were easy to grow, had a short generation time and exhibited clear distinct traits. Because for experimentation he can't use humans or other animals because it was too time consuming for the reproduction and pollination kind of thing. So, that is why the pea plants were selected. And among this pea plant he selected 7 specific traits to study, that was seed shape, seed color, the flower color, flower position, plant height, pod shape and pod color. All had distinct characteristics, and the presence of allele of for these characteristics, it was very clearly visible to identify the differentiation between the generations. So, this is an example where different kind of seeds were used, pods, flower colors have been used, where seed color may be a cotlet may be yellow or green, or flower was white or purple in some breeds, and then there was distinction between pods, and even the size of the stem was, in some cases it was large, and for other variations it was small or short. So, studying these characteristics was quite easy, and that is why the P was included, where he selected actually 14 true breeding pea plants for each trait as pairs, which were similar except for one particular characteristics. For example, he selected pea plant with all the variations common, but just one size, some were large and other were small in terms of their stem size. So, this is how he studied the trait of the size of the plant. So, rest everything was common in all these. So, the first step he did in his experimentation was the monohybrid cross. The monohybrid cross was at the first stage cross, which only one characteristic was considered at one particular time. See, because this was the first time that was being conducted, so he wanted to study at the very initial stages, he just wanted to consider one trait, and that was he crossed between tall and the dwarf plant. The size of the stem was the trait that he considered, which was the first step he took to analyze the different traits. He called these initially he considered all these traits, or all these things were associated with some kind of factor. He used the term factor, which was differentiating between the two plants, or that was being passed from one generation to another generation. So, he made a cross between a pure tall and a pure dwarf plant, pea plant. So, he represented with TT and with capital T and small t, because we are this plant also as humans, it is a diploid plant. So, we get two chromosomes from both our parents. So, that is the alleles are, so the genes are two. So, they exist in pairs. So, representation must be done in double form. So, the pure line is represented by the same symbol, and the difference between the two has been shown through the size or the capitalization of the term. So, he obtained all tall hybrid plants in the filial 1 progeny or F1 generation. The first generation that is created after self pollination, or the pollination of the not self pollination, but the cross pollination of the plants, it is known as the F1 generation. So, on self pollination of these hybrid plants produced all tall and dwarf in the ratio of 3:1 as the filial 2 progeny or F2 generation. When he pollinated the hybrid plants that he got as a F1, he obtained all tall plants, and in the filial 1 generation he crossed all those F1 generation through self pollination, and what he got as a filial 2 progeny or F2 generation was 3 tall plants and 1 dwarf plant, that is in a ratio of 3:1. This was the phenotypic ratio, that is 3 were tall and 1 was small. This is the phenotypic makeup, whereas the genotypic ratio or genotypic makeup of these plants were in a ratio of 1:2:1, where the first one is the pure tall plant, the second was with capital T and T as a genotype, and the last one is with small double t. So, it is a single cross between two organisms of a species that is made to study the inheritance of a single pair of genes or factors, because this was Mendel who used the term factors, because till that time gene term was not into the science terminology. So, he used this term as factors, which was passing from one species to another as a part of inheritance in a single pair of genes. So, this is how we can see the monohybrid cross, where capital T as representation, and for small t when crossed together, the genotype makeup that we received after first filial generation, that progeny that was created was capital T and small t. But then when this capital T and small t generation was crossbreed, and this was the thing that we received as the genotype was 1:2:1, that is, one was pure capital T, that is the long stem plant. The rest two were, although they were large stem pea plants, and one was with dwarf pea plant. The genotype was in the ratio of 1:2:1 that we can see, but if we see the phenotype, it was 3 tall plants and 1 dwarf plant. Why there was a difference in the phenotype, even there was 1 small t and 1 capital T in the middle two genotypic plants, it was due to the presence of dominant or a recessive gene. There are two kinds of genes. One is dominant gene, a gene that expresses itself in a presence of a contrasting gene in a hybrid. So, these two, if we can see at the last of the figure, the middle two are hybrid plants or hybrid generations, but still in the dominant one is expressing itself, which is the capital T is expressing itself. That is why the plant was, the stem was long compared to the recessive gene, which was represented as a small t, which was suppressed due to the presence of dominant gene. So, a recessive gene is that whose expression is suppressed in the presence of a dominant gene. In a hybrid, capital T and small presented as capital T and T, tall plant, the small t gene for dwarfness is recessive and the capital gene is for tallness is dominant. We can see in this figure at the very initial stage or the first side of the figure, we can see that the tall plant has the allele as capital T and capital T. So, that is why how we can understand that the capital T is a dominant gene, whereas a small t is a recessive gene. So, in presence of capital T or a dominant gene, the recessive gene is suppressed. That is why in case of hybrid, the small t has been suppressed and capital T is dominant. So, the phenotypes of the two hybrid alleles also is towards the longer side of the pea plant stem. So, this was the example. What we saw in the last slide was a monohybrid cross when only one character or one trait was studied, whereas now Mendel extended his experiments to reach to a dihybrid cross where two different traits has been considered. So, if we are studying two different traits, then 4 genes needs to be considered. So, they have been presented by two different alphabets, that is Y and R. So, this is a Punnett square which is finally showing that the F1 progeny that would be there would be capital Y, small y, capital R and small r. This is the first progeny that would be, or the first filial generation that would be developed after cross pollination. Whereas, when we consider the F2 generation, then the phenotypic makeup would be 9:3:3:1. We will see what all this mean and how it happened when we are considering two different traits. So, in a dihybrid trait as cross, that we initially understood that it is between two different traits that we are studying simultaneously in a different experiment. So, it is a cross between two individuals taking two contrasting traits and study the inheritance of two pairs of allele. And the genotypic ratio in the F2 generation is 1:2:2:4:1:2:1:2:1. And the phenotypic ratio in the F2 generation would be quite simple, that is 9:3:3:1. Again, the ratio has been different because of the expression of the dominant and the recessive genes, how they are expressing themselves. And again, I am highlighting that point, that studying the phenotypic ratio or looking at the phenotype of a plant or an organism, you cannot judge genotypic makeup of that particular organism. So, these two are not interchangeable or they are interrelatable because they are presenting themselves in such a way, but you cannot guess or you cannot define the genotypic makeup of any organism looking at its phenotypic makeup. So, this is a cross helps to study the principle of independent assortment given by Mendel. We will see how what 3 laws that were given by Mendel and how each experiment has supported that particular law. For example, the cross between pea plants having yellow wrinkled seed. See, this is how we are studying two different traits. One is the color of the seed and another is the shape of the seed. It is not round, it is wrinkled. So, we are studying two different traits, yellow wrinkled seeds and those having green and round seeds is a dihybrid cross. So, the two factors that we studied through last experiment that we shown was yellow color of the seed and the shape of the seed, that is wrinkled or round. So, now conducting all these experiments, Mendel finally reached to 3 different laws of genetics or inheritance. So, these were law of dominance, law of independent assortment and law of segregation. So, these 3 different laws were the baseline to understand about the inheritance or hereditary. So, after which we actually got lot of understanding of genetics and how traits are passed from one parent to another, and what are the differentiations between the phenotypic changes and the genotypic changes that occur across the generations. So, the law of dominance. So, law of dominance actually states that expression of only one of the forms of the parental trait in the F1 hybrid and in heterozygous conditions, as we have already seen what happens in a heterozygous conditions, that is the different alleles, the dominant allele gets expressed, that we have seen in terms of monohybrid cross where two different forms were used and even the genotype was hybrid, still the phenotype was based on the presence of dominant gene. The recessive gene was suppressed due to the presence of dominant. So, law of dominance is based on that experiment, that only one is dominant and will be expressed when two different alleles are present. F1 generation expresses dominant alleles. The suppressed gene is known as the recessive allele or trait. When F1 generation was created in that particular experiment where the size of stem was studied, then F1 generation was hybrid, was heterozygous in terms of allele, but still all the offsprings were of the taller size, because all the progeny was that was developed was capital T small t, but because capital T was a dominant allele, that is why all the plants were tall when we saw the F1 generation. Next comes the second law of Mendel, that is law of segregation, and in this, for any trait, each pair of allele of a gene segregate and one gene passes from each parent to an offspring. It is not that half gene from one chromosome and half gene from another chromosome will pass from one generation to another. It is that one gene completely passes from each parent to an offspring, that is the chromosome separates during the process of cell division and then a gamete is formed with one particular gene from one parent, and two alleles do not mix when they come together in a hybrid pair and are independent of each other. There is no mixing of these genes happens or these alleles happens, and these alleles do not show any blending, and that both the characters are recovered as such in the F2 generation, though one of these is not seen in F1 generation, but these do not show any kind of blending. And a homozygous parent produces all the gametes that are similar, while a heterozygous one produces two kinds of gametes, each having one allele with equal proportion. As we have seen, if the parent is double capital T, then the gametes would be one with capital T, another with capital T. There would be no mixing and the gametes would be similar. Whereas in heterozygous, the opposite condition would happen and two kinds of gamete, each having one allele with equal proportion, would be generated from that particular parent. So, we need to understand about the homozygous parent and the heterozygous parent, how the genes are transferred, why we say that in humans XY gene develops two different kind of gametes, and when they come in contact with the female gametes, that is both with X and X, that produces a different kind of embryo, because the heterozygous gamete was produced, kind of heterozygous gametes were produced. Thank you.